Hello everyone. The next topic which we will be discussing here is about the enzyme kinetics. So before going into the details of enzyme kinetics, we will first understand the basic concept of the enzyme. So enzymes, they are both proteins and biological catalysts. So that is why they are known as biocatalysts. What they do is they accelerate the specific chemical reactions in the biological system, and it alters the rate of reaction in the biological processes. Now, they are the enzymes, they are high molecular weight compounds, and they're made up of chains of amino acids which are linked together by the peptide bonds. Many enzymes, they require the presence of other compounds, for example cofactors, before their catalytic activity can be exerted. And there is enzyme kinetics, which is concerned with the influence of enzymes on the chemical rates. Now let's start with the introduction first, that what is enzyme kinetics. So if we talk about enzyme kinetics, then enzyme kinetics is nothing but a study of chemical reactions that are catalyzed by the enzymes. In enzyme kinetics, the reaction rate, the specific word here is the reaction rate, is measured, and they get how the changes in response to changes in experimental parameters, such as there is substrate concentration, there is enzyme concentration, etc. So the main focus here is the reaction rate which is being measured. The enzyme kinetics is a very oldest approach to understand the enzyme mechanism, and remains the most important. So it is one of the most important approach, and it has been used till decades. So it was being used earlier also. It is one of the oldest approach. So the initial rate is designated by V0 when S is much greater than the concentration of the enzyme, and there is a equation which is known as the Michael-Menten kinetics. It is the simplest and best known models of the enzyme kinetics. So the Michael-Menten equation is the rate equation for one substrate enzyme catalyzed reaction. So here is a graph which is representing the effect of substrate concentration on the enzyme kinetics. So the concentration of substrate is a key factor which is affecting the rate of the reaction which is being catalyzed by the enzyme. So as you can see here on the x-axis, there is a substrate concentration which is being denoted by the capital letter S, and this is the initial velocity. So here there is a graph which is being plotted between the substrate concentration and the initial velocity. So this graph is depicting the concentration, which is one of the key factor, and that affects the rate of the reaction which is being catalyzed by the enzyme. So here half V max, there is a Km which is being given, there is a V max which is also being given. Now the next we will talk about is the Michael-Menten's kinetics. So what does Michael-Menten's kinetics is? So Michael-Menten kinetics is one of the best known models of the enzyme kinetics. It is considered as one of the best models. So it was being named after a biochemist, that is Michael, and the Canadian physician which was Menten. So that is why the equation is named as the Michael-Menten kinetics. This model takes the form of an equation which describes the rate of enzymatic reaction by reaction rate relating reaction rate, the concentration of a substrate in its formula, and the formula is given as V equals to dP by dt, which is equals to V max, that is S Vm upon plus S. So this equation is the Michael-Menten equation. So here The Vmax, it represents the maximum rate achieved by the system happening at saturating substrate concentration. And the value for Km, that is, this is the constant, Michael constant, it is numerically equal to the substrate concentration at which the reaction rate is just half the Vmax. So the graph which I have shown you in the previous slide was nothing but only a depiction of the Michael-Menten equation. So this is the Michael-Menten equation. Now, if we talk about the applications of this equation, then the constant, that is KCAT, that is the catalytic, by Km, that is the catalytic efficiency, it is a measure of how efficiently an enzyme converts a substrate into the product. So this is the equation, and the application here is there is a diffusion limited enzyme such as fumarase which work at the theoretical upper limit, limited by diffusion of substrate into the active site. So Michael-Menten equation have been applied in a variety of spheres outside of the biochemical reaction. So it is not limited to the biochemical reactions only. It is outside the biochemical equations also, which is including alluvial clearance of dust, richness of the species in the pool, there is a clearance of blood alcohol, there is a bacterial phage infection. So this equation can also be used to describe the relationship between the ion channel conductivity and the ligand concentration. So these are some of the applications of the Michael and Menten equation. So this was all about the enzyme kinetics. These are all the references. Thank you.